Hello students, so good morning all. So today we will go for the next concept of electron transfer. In the last class we had seen that we had learned regarding the heavy and hemocyanide oxygen transport proteins in invertebrates, marine invertebrates. In today we will go for electron transfer protein. Okay? Electron transfer protein. So electron transfer protein, in that we will go for cytochromes. Okay, cytochromes. So they are found in plants. Okay, so they are found in plants and living in living systems. Okay, and these cytochromes, their work is to transfer the electrons. Okay, so they are the group of heme proteins. They are the group of heme proteins. They have porphyrin ring and they are classified into different types, that is cytochrome A, B, C and D. Okay, so they are classified into cytochrome A, cytochrome B, cytochrome C and cytochrome D. Okay, so depending on the structure, so they are having the heme structure like in hemoglobin and myoglobin, you have seen. So they are having the heme structure like this porphyrin ring, iron, and you are having the porphyrin ring here like this. Okay, so this is what we learned in the last class, and conjugation. So this is how the electrons, spiral ring of the heme group, that is porphyrin. Okay, and you can go for the conjugation now here. So means the ring go like this, like this, like this, like this. Okay, something like that. Okay, this is what conjugation you can see here, and this is the heme group of hemoglobin. Okay, like hemoglobin. So this is a heme group, porphyrin ring, with having the pylon of the nitrogen. Okay, and this is the iron, and this whole thing we call it as a porphyrin group, heme group. So this whole iron with the porphyrin ring, it is called as heme. This is called as the heme. Okay, and it is having the methyl group here like this. CH3, okay, CH3, CH3, okay, so CH3, CH3, okay, methyl group is there here in the heme group, followed by a long chain of CH2, CH2, CO minus, CH2, CH2, CO minus, okay, here CH double bond CH2. So like this, these substituents will make the heme group here. Okay, the hemoglobin, I had given you the porphyrin ring, but no substitution here for your simplicity, for the student simplicity. So directly I went for the fifth coordinated and the sixth coordinated for the hemoglobin. Okay, so fifth coordinated was linked by the nitrogen of histidine, and sixth coordinated you can see that oxygen is linked. But in this case, I have shown you the substituent, how the methyl group and this group CH2CO group long chain this alkenes group attached to the heme protein. So this is what totally we call it as a heme. Okay, so you can manage with the double bonds and all. I had said you 11 conjugated double bond, 11 double bonds will be there in the heme protein with iron as a center fit here. And now see here, cytochrome also has same thing, same feature, same structure here. That's why it is called as a group of heme protein. But what is changes here? Cytochrome A, B, C, D is there. So just a small change is there. Cytochrome A means what? There is a replacement of methyl group with formyl group, with something like this. Now, when one of the methyl group is replaced by formyl group here, so it is called as cytochrome A. So this is a cytochrome A. Is it right? Small changes in the substituents will give you cytochrome A, B, C, and D. Now in cytochrome B, what will happen? So this will not be there. Okay, this will not be there. Okay, and there is no attachment of protein group on this heme group. There is no attachment of the protein group on this heme group. So we call it as a cytochrome B. Cytochrome C, definitely there is attachment like this, covalent bond attachment will be there, and there is a protein group here. There is a protein group here, and this is attached by covalent bond. So when there is a protein group attached by covalent bond, then we call it as cytochrome C, and cytochrome D has a dihydroporphyrin ring. Okay, dihydroporphyrin ring. So if it is there, we call it as cytochrome D. So like this, for electron transfer, the cytochromes are classified into A, B, C, and D, and small difference between the small substitution of the methyl group. You can see that they are classified into different categories A, B, C, D, and their mode of transportation of electron is also different here. Okay, so and in cytochrome, so I will give you here. In cytochrome, in simple, so say diagram I will give cytochrome here. So iron is that all four sides is substituted by nitrogen here of pyrrole ring. Okay, so I will give like this. This is a pyrrole porphyrin ring, and here in fifth coordinated, so there is sulfur of lithium. Sulfur of lithium 9, and sixth is the nitrogen of histidine. So this is overall cytochrome, structure of cytochrome. See, that means there is no place here for cytochrome to attach the oxygen here, to attach for the oxygen. So that's why this is not directly influencing the oxygen carrying here, but indirectly it will reduce the oxygen. So it will transfer, it will help for transfer the electron and reduce oxygen, to water. Reduce oxygen to water, plus energy is liberated in the form of ATP. Plus energy is liberated in the form of adenosine triphosphate ATP. So this is how the role of cytochrome is. So they are mainly found in the mitochondria. Okay, they are found in, that's why they are found in the mitochondria of a cells, where maximum cytochromes are there, nearly 20 units of cytochromes are there, which helps in transportation of the electrons. And how do they transport the electrons? So you can see here how the cytochromes are transferring the electrons. Okay, they are transferring the electrons like this. So NADH to flavoprotein. Okay, then cytochrome B, cytochrome C, cytochrome A, then oxygen here, then this is reduced to water plus ATP. So this is how the electron is transferred like this. Is it right? So like this electron is transferred. So what is this NADH? So it is a reduced form of nicotinamide adenine diphosphate or dinucleotide. Okay, nicotinamide adenine dinucleotide reduced form. So it will give the electron, and that will be transferred in the cells, mitochondrial cells, with the help of flavoprotein, and then the cytochromes chains will be there. It will carry the electron, cytochrome B, then C, and mainly cytochrome A, which is holding the electrons and reducing oxygen to water molecule and etc. So this is what. Okay, so this is how the cytochromes are helping in carrying out what? In carrying out the transfer of the electrons, indirectly reducing the oxygen. And what happens here? Oxygen is reduced. Okay, and that energy is utilized for what? That energy. Okay, so oxidized. So oxygen will oxidize and gets reduced, and that energy is utilized to break down the carbohydrates. Okay, to break down the carbohydrates, and during this process, where the electrons is released during this breaking of carbohydrates, the electrons what are released is transported by whom? By this chain. Okay, and they are transporting these electrons from this chain like this, and finally they are reducing the oxygen to water molecule and generating the energy, that is adenosine triphosphate ATP molecule. So this is how you can see that the cytochromes play important role here. So in this case, one thing comes here: cyanide poisoning. They will ask this is cyanide. What does this mean by cyanide poisoning of cytochromes here? So in competitive exam also they may ask this. So what will happen when any person is injected by the cyanide? Here what will happen? Cyanide has great affinity to bind with cytochrome A. Okay. So when it binds with cytochrome A, what will happen? The electrons will not flow to oxygen. And there is no reduction of oxygen and no energy. The cell dies. Okay, the cell dies here, and that's why what will happen? So cytochrome A binds with cyanide. Okay, it binds with cyanide and they form the complex here, and there is no flow of electron. There is no flow of electron. No flow of electron here. Okay, so this is a case where the cell dies. That's called that is what called cyanide poisoning. Okay, then what we can do it for patients here. So those are injected with cyanide. So they are given the compounds like amyl nitrates. Okay, so they are given the compounds like amyl nitrates. Okay, what will happen? So they will oxidize this iron from this to iron 3. Okay, so amyl nitrates injections, which is given during cyanide poisoning. So what is that work? Their work is its work is to oxidize the iron 2 to iron 3. So what will happen? Iron 3 is what? Methemoglobin. Okay, so they are going to methemoglobin. So what will happen now? Iron 3. Now this cyanide, which is bound to cytochrome A, will detach and bind to this Fe+3. And like this from cytochrome A, the electron will now reduce oxygen to water molecule. And what happens to cyanide? Cyanide will bind this Fe+3 and they are passed to the urine. They are gone to the urine. This is how the treatment is done. Okay, this is how the treatment is done. If any of the patients is infected by the cyanide. Okay, like carbon monoxide. So what about carbon monoxide poisoning with hemoglobin? So when hemoglobin is there, so if carbon monoxide is inhaled by any of the patients, it is going to form carboxyhemoglobin. And oxygen binding is very, very less. Okay, 1000 times carbon monoxide can bind to hemoglobin. See, that's what poisoning we say. So more if you expose for the carbon monoxide, so that will lead to what? Sometimes death also. Why? Because whole the body system will be infected by the carbon monoxide with hemoglobin, forming carboxyhemoglobin, and oxygen transportation becomes very less toward the body. And what will happen? The patients may have the problems with the respiration, and sometimes death also happens. Okay, and that's why cigarette smokers and all, so always they are inhaling the carbon monoxide and they have the problem. So that's why that is the reason why we have to quit the smoking and all. So because carbon monoxide has 1000 times affinity for hemoglobin than the oxygen. Okay, and that's why. So this is what, but this is a reversible reaction. That's why the patients use, though they are exposed to carbon monoxide, and if they go to fresh air, again that will replace the oxygen. So this is how it goes here. But it is also very dangerous, carbon monoxide, forming the carboxyhemoglobin rather than oxyhemoglobin. This is a oxyhemoglobin. So once it comes in contact with carbon monoxide, it will replace this oxygen, forming carboxyhemoglobin. So that is how it is called as carbon monoxide poisoning. Like in cytochromes we call it as a cyanide poisoning. So in carbon monoxide we call it as a carbon monoxide poisoning. Okay, this is how. And this is what cytochrome C oxidase. Okay, one more cytochrome C oxidase. So its work is to reduce the oxygen. Okay, so its work is to reduce the oxygen. We taking the electrons, forming like this. Okay, so this is how the cytochrome C reduces. Cytochrome C oxidase. Okay, they are going to reduce the water molecule in this way. So cytochrome C, the cytochrome C oxidase, cytochrome A will hold this and it will go to here. So even cytochrome A and this also is have the more role for oxidizing the reducing the oxygen. So this is how I can say that the cytochromes play important role in transfer of the electrons from the reduced factor NADH to the oxygen, and then the energy packet is being released when oxygen is reduced to water molecule. Is it right? So this is how the cytochromes play important role and that's why in the photosynthesis also we see that chlorophyll. Okay, so there are a lot of cytochromes A, B, C, F. So when we study the chlorophyll, we will come to know. So these are the transporters, okay, electron transporters, and finally they are using the oxygen. Is it right? So this is how it's a group of heme protein. So they are having the porphyrin ring as we learned here. Okay. So iron is there, but there is no scope for oxygen to bind there because the fifth coordinated to sulfur of methionine and the sixth coordinated to the nitrogen of histidine. So that's why indirectly its work is to transfer the electrons from the after that it's breaking, and finally to reduce oxygen, finally to reduce the oxygen and reduce oxygen to water molecule. So this is how the cytochromes do play work in the living system as well as the most plant system, as I said, the chlorophyll. So in plant system also we will see this cytochromes electron transfer system. Okay, so hope you understood this cytochrome. So be specific for this. And cytochrome C oxidase, as I said, so cytochrome C oxidase, so they contain copper A and copper B as the metal ion. Okay, and these with heme iron. Okay, these with heme iron too, they will what? They will reduce oxygen. They will reduce oxygen or transfer the electron. So this is how in the computer examination you should know this. Okay, so cytochrome C oxidase, that is what they contain two base metals, that is copper A and copper B. So along with Fe+2 of heme, they help in transportation of the electrons and also for reducing the oxygen to water molecule. So like this cytochrome A, B, C, D, they are having different roles here. So mainly electron holding will be for cytochrome A, is it right? And that is what about the cytochromes. In the next video, we will go for the iron sulfur electron transfers. Okay, so that is also very important concept. Hope you understood. Have a nice day. Thank you.